So greetings and a very warm welcome to our audience joining us from around the world at this hour for the UCA's faculty lecture series. Today I have the pleasure and the honor to serve as a host and also for this lecture. Today's topic is management of soil and seed-borne diseases of potato, Solanum, tuberosum, sorry if I'm mispronouncing it, and Turnip, Brassica, rapa in Michigan. It will be delivered by my colleague, Dr. Saltanat Mambetova. So before we proceed, let me remind us of some ground rules. Please have your microphones muted when the lecture is in process. And I'd like you to not interrupt the speaker during the lecture and reserve your questions for the end. You may type in your questions in the chat box and I'll ask them at the end. Or if you prefer, you may raise your hand at the end of the talk and then ask your questions yourself. Okay, with that said, let me introduce our speaker. Dr. Saltanat Mambetova is an assistant professor at the Department of Earth and Environmental Sciences here at the UCA's Khorog campus in Tajikistan. Before joining UCA, Saltanat was a postdoctoral researcher at the Michigan State University, where she also received her both master's and PhD degrees. Her research interest is in agriculture, including food security, integrated pest management, agricultural biotechnology, and agricultural technologies. While she was at MSU, she was actively engaged in collaborative programs between MSU and Kyrgyzstan, working to build a cultural and agricultural technology bridge to Central Asia. Saltanat has also played a leadership role in transferring potato varieties and testing them in different geographic regions of Kyrgyzstan. With that said, I'd like to pass the mic to Saltanat. Saltanat, the floor is yours.
Thanks so much, Maratek, for the introduction, and thanks for being a moderator today. I also join you and welcome everyone who joined today's seminar. Thanks so much. With that, I guess I will start my talk. So today's my seminar will be, I will share my PhD / postdoctoral research work that I did at Michigan State University. And mainly, I'll be talking about two projects. One project is on evaluation of conventional polymerase chain reaction methods to detect Dickeya and Pectobacterium species. And another project would be on, I will be talking about genotypic and phenotypic characterization of Streptomyces species on potatoes and also on turnip. Before I go in and describe about these two projects, I would like to mention or give a general information about importance of potatoes and turnips as well. So potatoes, the most important non-grain food crop. And I'm emphasizing non-grain food crop because, you know, the three important ones, the wheat, rice, and corn, but the potato stands as a non-grain food crop in the number one. And potato production in the US has highest volume and compromises the largest harvested acreage of vegetables. And particularly in Michigan, potatoes rank 7th in the production in the whole country. And the estimated gate value is about $207 million US dollars. And another fact that I would like to share, all the potatoes that are grown in Michigan, 70% out of it goes into the chip production. And just want to kind of highlight it and tell you a very short story. When I arrived, first time to the United States, I was amazed and overwhelmed, I guess, with the different flavors of chips they had. And this is just one example that I'm showing. This is a local Michigan factory that produced potatoes, the Better Mates, and you can see, the varieties of different chip flavors are in the pictures. And also, I was, lucky enough to visit the factory, and I've seen the whole process how, the potatoes turn into the chips. While at the same, when I, about 10 years ago, when I arrived to United States first time, I was overwhelmed with the chips because at that time, at least in Bishkek, where I lived, we only had, very, few options, and potato chips were not as common at that time. So, but now, of course, after 10 years, the potatoes, changed, I guess, in Bishkek and overall in Central Asia. It's, there's lots of potato chips, potato wedges. You go to restaurants, you can order easily, potato French fries and so forth. So things obviously have changed since then, but I kind of wanted to give you a very brief chip introduction. The second crop that I would be talking today, it's the turnip, and turnip production in the US, it's ranked number 4 and worldwide. And in Michigan, the turnip production is ranked number 1 among the other states. And lately, I guess between 1997 and 2012, there's almost 200% of increase of turnip production. And that's, I guess, one explanation is how people are using turnip in a different variety of ways. And I guess if you're wondering how, what turnips be, or how you can eat them and use them, again, in the States, I've seen pickled turnips are very common, and also fresh turnips are common, using, when cooking in a different cuisine, and also fresh leaves, the top part of the turnips are also very common. So these two crops that I will be talking about, and I guess the diseases that get affected by these two crops. So first I will go through project 1. And as I said, it is an evaluation of conventional polymerase chain reaction methods to detect Pectobacterium and Dickeya species. And just overall, I'm gonna give you overall information about the disease. So as you can see, the name is long. It's blackleg aerial stem rot and tuber soft rot of potato. And in a couple of slides, I will be sharing the symptoms of the disease, and it will make sense where the name came from, I guess. So these diseases caused by Dickeya and Pectobacterium species, they are known as seed-borne. This is an important disease in UK and Europe, Israel, and now it's in the US. And I guess a little bit of a story behind how it became important. Back in 2015, there was a Dickeya outbreak in North America, and the picture that you see in your screen, it's the potato field where you see a lot of gaps within the rows and also within between the plants, and this happened because of the this disease has been caused this outbreak. So it is very it's a very sad picture, because growers would like to see full plants here where you see empty spaces. And similar story happened in Michigan as well. So we've, growers have called, different researchers and extension services and share these pictures where, again, you see empty, rows and spaces where potatoes is supposed to be grown. And this was, happened, the summer of 2015, and our lab has published first report on this disease, and we found 2 new species, Dickeya and Dickeya and Pectobacterium wasabiae, that caused aerial stem rot in Michigan, and we published a report on it. And following years, like following 2016 spring and summer, the disease has been spread all over the states, like Maine also published, and there were many other states that potato production has been, the disease reached to those potato production states. And not only in the states, these new species were discovered also in Europe, and there were a lot of broadcasting and reports have been out there. This is one example out of Netherlands. Another one, it was in Australia as well. where they found this new species of Dickeya and Pectobacterium being spreading around. So the bacteria has been spreading not only in the US, as I said, but in worldwide. And one, I guess, explanation kind of the outbreak of in the US, it was that the Dickeya probably been in the seedlots, the potato seedlots, for a few years. And then in 2013 and 14, there was the rain probably took over. I mean, the rain spread the pathogen, but cool temperatures kind of kept the caused the latency, because in 2015 the spring was very warm and temperature raised, and the outbreak, we at least several states in 2015 saw a significant outbreak in 2015. So this is kind of an explanation why it all of a sudden emerged in 2015, this disease. So this disease, there are different species that cause these blackleg, aerial stem rot, and tuber soft rot disease on potatoes. And there are multiple species of Pectobacterium and Dickeya. So here you see on the screen there's 5 at least different species and subspecies. And also there is the same thing. There are multiple Dickeya species that are present in US.
Wow, long time, my friend, very long time.
I hope you will.
It got a little bit disrupted. So the multiple Dickeya species also being present probably in US seed production, but the Dickeya and Dickeya, which appeared that year was the common and the most damaging one. And among the Dickeya species, Dickeya solani is very aggressive, and one of the aggressive species among Dickeya bacteria, but it's not yet reported in North America, but it's been reported out in Europe, Netherlands. It emerged in Netherlands, and it's been found in other European countries. So before I'll go and explain the methods and methodology, I would like to share the disease cycle, because it kind of helps to see how disease occurs and spreads, and how from the management side, how you can manage the disease, and where it's important to understand the disease. You need to understand the disease cycle. So this is disease cycle of these tuber soft rot, blackleg, and aerial stem rot. So I'll just briefly explain. I'm not going to go into details, but you have potato seeds and that, let's say, have some inoculum or bacteria infected. So you plant them in early spring, and during the planting, usually again in the United States, seeds get cutted. So while cutting and transporting, you can easily spread the disease. So the disease can be spread while you're cutting and preparing for the planting, and then the disease will spread from infected tubers into the stems, like you see here in the pictures. And in the field, you will see these blackleg symptoms or aerial stem symptoms of the disease. So with, again, the rain splash, irrigation splash will spread the bacteria more to other plants, and you will end up with infected tubers. And once you harvest them and store them, then next year that infected tuber would be your inoculum, would be your primary inoculum, and again you will be spreading the disease again and again, and the disease cycle continues on. So it is, again, important from the management part, and I'm gonna mention it in a couple of slides. I'm just gonna show you a couple of symptoms that are real picture symptoms. So these are some examples. So kind of early examples of this disease are upper leaf curling and wilting. However, this is very common in other diseases as well. So this will not be your specific symptoms of the disease, but it can include upper leaf curling and wilting. The more specific symptoms are, as I said, are in the stem. So where the potato plant's growing, you can see in the pictures with the blue arrows and the circle, you can see the necrotic tissue, or that tissue right here. And once you cut the stem, you will see that necrotic tissue. So basically, these bacteria is spreading upward and onward. It can spread through the stem and also to the tubers downward. And here are more pictures. So this is, again, I think the name of the disease will explain again, the aerial stem rot. Right here, you see the whole plant get infected. There's, these necrotic tissues or dead tissues. And then here from the seed perspective, you can see, the diseased tuber was planted, and you can see how this bacteria is spreading over to the stem, and also it's causing, the soft rot in the tuber. So the soft rot name, it comes from the tuber because it will make the potato tissue very soft and mushy, because the bacteria produces pectinases that break down the pectin in the potato tuber. So early detection is the key. So first step to, in effective disease management is to identify and make sure you have, accurate identification and diagnosis of your disease. So as you can see from the slides that I just showed you from the symptoms, Dickeya and Pectobacterium species, they are often indistinguishable from each other. So in the field, it will be very difficult to see these two different species on the potatoes and then say, oh, this is Dickeya caused, or this is Pectobacterium caused. It's kind of impossible. I mean, there are some certain, recommendations you can go through, but it's very indistinguishable. So accurate and sensitive molecular diagnostic are essential to detect these two species, and particularly in seed certification, because as I showed you in disease life cycle, the seeds can be the tuber seed can get infected, and it can be primary inoculum next year when you plant that seed. So it's very important to detect the disease. So the research objective of this first part was evaluate the fidelity of available PCR primers to detect these two species, Dickeya and Pectobacterium. So samples for this project were collected from various Michigan growers, and there was 203 symptomatic samples. And when I say symptomatic, these means the symptoms, the plants or potato tuber had actually symptoms like these I showed you in the picture, or in actual potato tuber. This was actual potato tuber, but it got smooshed. As I said, the bacteria makes this tissue soft, and that's why it doesn't look like anymore a potato tuber. And then also in this study we collected non-symptomatic tuber samples from seed lots, and mainly these are just the seeds, normal seeds that you don't see any symptoms, and then you collected and brought to the lab and then tested them out. So for the isolation part, I'm not going to go into details, but just briefly we'll explain it. So we divided, again, symptomatic and asymptomatic samples, since we had two different ones, and basically tissue from both either a stem or from a tuber being isolated and following lab procedures, and then being plated on this media called CVP. It's a crystal violet pectate medium, where basically, again, we would see these once you plate these bacteria, and if it is a Dickeya or Pectobacterium, it will produce these pectinases and break down in the media. You will see these little pits. So once you get these little pits, then you can re-isolate. And this is selective media, so we're selecting for this specific two bacteria. And then, of course, once we get the pure culture, DNA extraction was being done from the pure cultures. And from non-symptomatic samples, it was similar, a little bit different methods, but basically a tuber, from the non-symptomatic tuber, a tissue core has been taken from it, and it was grown in a, again, specific selective media. And once we grow out the culture, the DNA extraction was done from these particular samples as well. So basically, all the samples that we collected, they were plated on CDP agars, both symptomatic and non-symptomatic ones. All the samples being tested via conventional PCR using different primers, and I will talk about primers in just a minute. And also 10 samples being sequenced using 16 RNA gene to confirm and identify also in the sequence level what species of bacteria these in the samples we had. So this is a list of PCR primers. So just want to, I guess, mention also prior to 2015 outbreak, there were already, research on both these bacterial species, but since, there was no outbreak, there was no emergency, I guess, of using primers. But when the outbreak hit and, growers were really needed to identify and know what bacteria they have, they really, kind of pushed the research, and we had to select PCR primers that were already available and published in the published, papers. So basically, we went through and selected the ones that we thought that would be, best suitable ones, and these were the list of these different PCR primers. Now, I'm not going to go through, but kind of giving you, overall an idea, right? And we had conventional, so real time, but today in my talk, I'm only talking about conventional PCR primers. So regarding the field sample results, they are mostly symptomatic ones. All of the isolates that we screened were confirmed that they were pectolytic on CDP agar, that I just mentioned, the selective agar. And the sequence, the subsamples of these samples were identified as Dickeya and Pectobacterium, Pectobacterium cartavarum, and Pectobacterium parmentieri. So there are 3 species, so 2 from Pectobacterium and 1 from Dickeya. And so from the PCR primer list that I showed you, we identified 3 of them that suited best and were more or less accurate and reliable. So for Dickeya and Dickeya, we choose this PLAD primer, and out of samples that we tested, 62% came out positive for this species. And then EXPCC was for 2 Pectobacterium species, cartavarum and parmentieri. 6% of the samples that we tested came out positive. And another primer was Y45 and 46. And this identified Pectobacterium atrosepticum, which were 2% out of the samples that we collected. And there was 30% of negative for all assays. And in the seed lots, that non-symptomatic samples that we collected, we used the same PCR primers, and you can see there was 0 for Dickeya, there was 23% for cartavarum and parmentieri, and 6% for atrosepticum, and 71% of the tested samples were negative. So in the conclusion of this study, I mean, we had to, again, using the list that I showed you, use all the primers and identify them, which ones were reliable and accurate to identify up to species these 2 bacteria. And what we found at the end, the PLAD was specific for Dickeya and Dickeya. However, it can also sometimes, so it was not as specific. So with Dickeya and Dickeya, you can use this primer to identify, but it was also identifying other species. However, the EXPCC was specific for these 2 species, cartavarum and parmentieri, consistently, and Y45-46 species primers was specific for Pectobacterium atrosepticum. So this is kind of the conclusion of one of these, I guess, the conventional PCR primers part. And I'm moving to my second project, which is genotypic and phenotypic characterization of Streptomyces species. And now I'm going to talk about both potato and turnip. So a little bit about, I guess, information about the disease itself. It's called common scab on potatoes and scab on turnips, I guess. It is another bacteria. It's a gram-positive bacteria. There are about 500 Streptomyces species. However, only a little amount, maybe less than 13 species are pathogenic. And most of them are non-pathogenic, at least 2 plant species. And it's very common bacteria that is found in the soil and on decaying vegetation. It's after rain, when it smells that earthy smell, it is because of these, I guess these Streptomyces are responsible for that smell because they produce jasmine enzyme that the chemical that gives this earthy smell. And as you can just see in the picture of these, turnip and potatoes, it is, caused a great significant losses. And in potatoes, I'll show you in the next picture, these scab lesions on potatoes can be very deep, and it will not be able, for any fresh market or cheap production, you will not be able to use it because of these very deep pits. And in turnips, it also caused these lesions, as you can see here, and it's also reduced the marketability. Again, just briefly will explain the disease cycle of this bacteria, the Streptomyces. And again, we can start, I guess, from this winter to spring. If you have, potatoes that is infected with this bacteria and placed, and planted. And one thing with this bacteria, Streptomyces, it's a kind of unique bacteria. It produces hyphae. Usual bacteria do not produce hyphae, and this bacteria produces hyphae, and these filamentous grows. And on the plates, when you grow them, they have these filamentous grows, like mycelia, like fungi grows. But it is not a fungi, it is a bacteria. They produce these spores, and spores infect the potato tubers. And again, the rain, irrigation may spread the bacterium from one plant to another plant. And what happened during the season, these bacteria germinates, and they start infecting and go through under the potato tuber and start making these corky layers. And you can see in the picture here, it's showing how these bacteria spores grows under the skin, and it starts making these lesions. And that, as deeper it goes, it will create deeper pits, and if it stays kind of on the surface, then it will make that surface lesion. And again, if the potatoes, harvested and stored, and it can serve as a primary inoculum next year if you plant infected potato seeds. So here's just again want to show you the severity of the disease. So in my research, I selected different type of lesions. So I'm just going to kind of briefly mention this here in the pictures so you have idea. But the pictures, from A through F is just explaining how this disease can be severe on potato tubers. And so on the A, you see it's very superficial, like discrete lesions. And in B, it goes, these superficial discrete lesions coalesce and makes a larger area or infects larger area. In the C, you see it gets raised. These lesions get raised right here. My mouse is not working well. And in the D, it's get these raised lesions get again coalesced, makes a big infects bigger lesions. And then E, where it makes pit or these little holes into the tubers. And then in F, it's again the coalesced pitted lesions on the tubers. So, it is very, very important. And as you can see, the disease can, make from A through F. It's, it changed the actual potato. This this little potato doesn't even look like potato anymore. It's kind of has all these holes on it. And on turnips, at least what I was doing my research, we didn't see this, severe pitted on turnips, but we did see other symptoms as well. There was a pitted pitting on or deep holes on it, and also superficial lesions as well. So the objectives of this research, one was to collect pathogenic Streptomyces species that cause the scab, both from potatoes and turnips. And number two was characterize phenotypically the virulence and pathogenicity of potatoes and turnips. Number three was characterize pathogenicity factors using PCR primers. And number four was determine the diversity of Streptomyces species associated with scab on both potatoes and turnips in Michigan, and understand their phylogenetic relationships, how they're influenced by phenotype. So with this one, we also collected samples from various growers from Michigan for both potatoes and turnips. And again here, as I showed you these symptoms, we divided them from pitted versus surface lesions, and for both turnips and potatoes. And I think turnips in this particular study didn't have any pitted lesions, but had only surface lesions, but potatoes did have both pitted and surface lesions. So in total, there were 51 isolates, and pathogenicity assay and phylogenetic characterization were done on these 51 isolates. So first, we done the morphological characteristics, where we looked at the the host and lesion type. We looked at the superficial turnips, superficial potato isolates, and also pitted potato isolates. We look at the mycelium and also pigments, because Streptomyces produce different pigments, like red, sometimes reddish, pinkish, and buff red, and other pigments. So we looked at that, and then we also did two pathogenicity assays. So one was a tuber disc assay, and one was turnip seedling assay. And again, briefly, in the tuber disc assay, basically tuber discs were made up from the potato tissue, as you can see in the pictures. Then they've been plated on the plates, Petri plates, and the Streptomyces, different isolates of Streptomyces being placed on top of the tuber disc, and pathogenicity or severity of these pathogen being, recorded and observed. And then the second, with the turnip, we did turnip seedling assay, where basically identified a turnip variety, and we plate them on these again Petri plates on medium, and under the once the plants grew a little bit, then we strip the different Streptomyces species under the seedlings, and then we recorded measured the length of the seedling, and we looked at how much each seedling grew. And of course, both tuber disc and also turnip seedling assays being replicated 3 times, and as you see, there's 5 seedlings per plate, and there was 5 tuber disc per plate. And then once the DNA has been extracted from the mycelial growth of these isolates, and then using different different pathogenicity gene primers being used to identify pathogenicity, and 16S was used as a reference gene locus. So for the DNA sequencing, there were we used the same primers that we use for conventional PCR, and for phylogenetic analysis, we only used 16S RNA, RAC-A, and RPLB locus. And genetic analysis were, the phylogenetic analysis were done using Bayesian methods using POP software. So in the results for pathogenicity assay or seedling assay, we didn't see or we didn't find any significant differences among isolates based on pathogenicity seedling test. There were differences in or differential responses based on host and tuber disc assay of all of the Streptomyces isolates. And among, I guess I'm missing one. Okay, among 20 Streptomyces isolates that were from potatoes, 17 found necrotic, and among Streptomyces isolates that were from turnips, out of 23 were necrotic, and then from the superficial isolates, among 11 ones, there were 1 necrotic ones. And in the phylogenetic trees, we for the first, I'm going to go for the PCR results. So we used 3 genes, the textomin A and textomin AB and NEC1 and TOMA. And these 3 are genes are required for pathogenicity, or the Streptomyces species needs to have these basically regions in order to be pathogenic on these potatoes and turnips and other crops. So basically, we found you can see numbers out of 20 superficial turnip isolates. There were 7 that had textomin A and B, 3 had NEC1, and 17 had TOM-A. And then from the pitted scab lesions, there were 17 had higher numbers had textomin A and B, 17 found NEC1, and 20 out of them had TOM-A. And for superficial last one, 3 of them had textomin A and B, 3 of them had NEC1, 7 of them had TOM-A genes. And with the all 3 phylogenetic analysis, this is the 16S RNA gene. Here, basically, and it is similar with the other 2 trees, that Streptomyces species that had superficial turnip and superficial from turnips and potatoes, the lesion types grouped together. The pitted lesions from potatoes grouped together, and same thing, kind of similar trend we saw with the RNA RPOB or RNA polymerase B subunit gene as well, where superficial turnip isolates grouped together again. As you can see, the superficial turnip and potato isolates grouped together, the pitted isolates grouped together, and these were the referenced species that we used from public data set. So in the results was, again, with the phylogenetic parts, all these 3 phylogenetic trees show a very similar trend. The superficial isolates clustered together in all analysis, the pitted isolates from potatoes clustered in all analysis, and superficial isolates both from potatoes and the turnips clustered together. And in the 16S RNA, we showed the pitted isolates also clustered with the reference pathogenic Streptomyces species. And in the summary, out of all the 51, we successfully were able to isolate 51 isolates total, 20 from turnips, 20 from pitted potato lesions, and 11 from superficial potato lesions. Turnip and potato isolates, they all showed the suppression in the seedling pathogenicity assay. However, we didn't see any statistical significance. And 17 pitted, 3 superficial potato isolates, and 3 turnip necrotic isolates being identified, and tuber disc assay. Most isolates from pitted lesions contain both textomin A and textomin AB genes. Most of the superficial isolates that were from the surface lesions didn't have NEC1 gene, which is kind of very surprising because NEC1 is also one of the responsible genes for pathogenicity. And most of the isolates from turnips and potatoes had TOM-A gene that is also responsible for pathogenicity. And in the phylogenetic trees, again, the potato isolates from pitted lesions, in all three phylogenetic topologies, clustered very similarly, and also, the reference pathogenic Streptomyces species also clustered with the pitted ones. And I guess, so then the isolates also, from the superficial lesions also had combined with the potato ones. So, which basically what we think is the superficial isolates that caused, both on potatoes and turnips have similar genes versus pitted ones. That's why we did see this segregation. And I guess that said, I tried to be on time, and I guess I went a little bit over. But thank you, and I'm ready to take any questions if you have any.
Yeah, thank you so much, Saltanat. You are perfectly on time because we started late. So thanks for a very informative and engaging talk. Don't questions or raised hands so far, but let me kick off the Q&A session with a question of my own. This is not going to be much about the genetics and other aspects, but I'm having, been growing potatoes in my farm with my granddad and parents, and the tradition still continues. The pictures that you showed of these different kinds of diseases looked very familiar, although I wouldn't be able to name which one is which. I guess the question that I have is, we do have these kinds of issues here, and with the lack of sophisticated lab facilities. Do you think is there a potential to sort of provide the farmers with some, kind of very basic tools, I don't know, techniques to be able to identify these diseases so that they can avoid propagating them in the next cycle of the sowing and the harvest?
A very good question, Wojtek. And I guess you're touching a very good or important issue. In order to, I guess we can help farmers identify diseases, by, I guess, visual, looking at symptoms and things like that. But again, to give a proper identification, it would be best to use some type of lab facility where you can, grow out and do, if not like genetic part, but at least morphology and looking at, spores and things like that. But in overall, give a general information, yes, it's probably possible. And actually, one of the NGO here in Kyrgyzstan developed these apps on phone. Now everyone has smartphones, right? So they developed this app where they downloaded, and this NGO is very, very close with growers here in Kyrgyzstan, and they help them to identify. But basically in the app, they can see these pictures and they can kind of compare to their own diseases and then tell. But sometimes it can get tricky, especially when if you're looking at the foliage of the potato, the leaves, then some diseases can have very similar symptoms. So it is kind of hard, but I guess my general, or I guess my goal is actually, hopefully, at the UCA will be able to provide them this type of, we'll hopefully open the facility where we can identify some of the disease and disseminate the research and tell them what kind of disease is common in the area. So I guess hopefully, did I answer your question?
Yeah, absolutely. This is something that we would be really providing an excellent service to the growers around here. So hopefully with the PCR coming in other techs, the lab will be up and running. And so we'll look forward to some fantastic results there. All right. So,
Exactly.
thanks again. And let me move on to the chat. I see a couple of questions here. So Salmaz, would you like to read it yourself, or would you like me to read the question? If you would like to read it yourself, just feel free to unmute and ask the question.
Yeah, not a problem. Thank you, Saltanat, for this great talk. I'm just wondering, you mentioned at the very beginning, and this is related to also what Outbreak asked you, so probably you've already answered this question, but I'm more interested in the seeds, because you said that there was a presence of some of these pathogens in the seeds for several years before the farmers knew about them. And so is there any tool or anything that can be used systematically to test these seeds before, they become so problematic? If they have the opportunity to be present in a, for the pathogen to be there for, several years before it actually becomes a problem?
I mean, there's two parts, I guess I would say. If it's a general question, almost right, not in Central Asia. Is it a general? Just want to double check.
This is a general question, and okay. Specifically for the example that you used in your talk.
Okay. Yes, as a general. That's why I guess one thing. I guess it's very common, I would say, with the growers, and I guess I mean, there are some participants here maybe can add if I'm not wrong, that Professor Georgi, so he was in the participant list. But basically, when that Dickeya, the pathogen hit the United States, with the outbreak has happened. Usually, farmers, they grow their crops, they're happy until something happens to them, and then they kind of freak out and they're like, okay, the disease is here, so what can we do? How can we eliminate, right? But the researchers, we always do a research. So the good thing was the research already been done, and that's why we already had some kind of information, and we selected these PCR primers in order to identify them. So there was some research being done already. But the second thing, that's why once these outbreak happened, then the question was, okay, how are we gonna make sure that, like you said, the seeds are clean, that we're not spreading everywhere and across the states. And that's why where the PCR primers came and the new technology, I mean, I only talked about the conventional PCR. There's also qPCR, the real-time PCR, and the continuation of this project is to identify actually even quicker and reliable. There's different tools that you can actually take it to the storage. They made this lamp. They call it's like a little PCR machine, but it's you can carry it in your bag. There's a carry-on technology. Basically, you would take the tissue and start. You can do the process in the where you can take in the field to the storage. And that's how you can identify. But they were not, they didn't have primers for that. So they were developing and working on. The one thing about this particular disease with Dickeya and Pectobacterium, the genome of these two species are very similar. I think there's 97 or 99% of genome is very similar. And it's very hard to make these specific primers. And especially in such carry-on techniques, they don't use a very high throughout technology, high throughput technology. So they were working on identifying a very specific primers where you can identify. But unfortunately, I guess with seeds, it's kind of, especially with asymptomatic ones where you don't see the symptoms on the seeds, it's hard to identify. In the US, there's a seed certification programs where they have certain diseases, not this, but other diseases like viruses and nematodes that actually are prohibited. Like there's 0% of having that, disease in your seed lot. So they do test those. There's a seed lot testing happens, but not with this specific disease. So there is that things happens in the US, but we don't have in Central Asia. We don't have a seed certification programs at all. I hope I answered your question.
All right, thanks very much. There are a couple of comments in the chat. I'm going to just read them in case you want to read them yourself. And then we have a couple of hands raised, so we move on to there. David Rogers is saying here in Idaho, we are starting to use drones to identify and map potato crop damage in large fields. And Sadria is saying, yes, that is a very good idea. And I would say that this is what our farmers in Gbao need to know and get some kind of support. So I guess this is agreeing to what you answered the question about my question in providing local farmers with the support.
I just want to add in Michigan as well, they use drones, just commenting on David's Rogers comment. They also, I mean, used very widely to identify disease, especially again in the US, the acres of the potato fields are huge, you know, you can't just walk through them. And they use this technology to identify, especially, you know, during the growing season, and not only for diseases, for nutrient issues, for many other issues that you can, you know, fly the drone, look at the pictures, and then go specifically to that, you know, to that location and identify what's going on with your plant. So yes, it is very common. Okay. And thanks, Adria. Hopefully, Adria will be working together, and we'll help Khorog and Pamir region, I guess.
Sorry, Saltanat.
Yes, Saltanat, we are waiting for you.
Add a comment to that as well.
Yes, Ben. Yes.
Hi, everyone. Just add to that interesting topic of using drone in agricultural practices. Yes, we are working on that. We already bought 2 drones, which specifically can be used for agricultural practices. They are multi-band, small remote sensing camera, which can be used to detect different disease by flying those drones above the fields. We have 2 issues to deal with. One is being very close to the borderline. We are working to have a permission to be able to fly those drones in Tajikistan. And the second issue, I guess, being located in Khorog and not having even a degree or certificate related to agricultural practices, that's something we need to work at UCA to develop some courses or certificate courses or training workshops, basically to transfer that knowledge to local farmers. As Muradbek mentioned, they've been growing potatoes for many years with local knowledge, but it will be good to have some certificate courses or training and workshops, you know, to deliver the new technology and new knowledge to them. Thank you very much.
Well, thanks, Ben, for the input and clarification. It's definitely exciting to have some drones, and hopefully we'll be able to operate them. All right, now moving to the part where the hands are raised. So I'm going to go by the order that they raised. So first up is Amrain, if I'm pronouncing it correctly. If I'm not, my apologies. Please, I'm muting. Go ahead and ask a question.
It's okay. It's Amrain. Obviously, Saltanat, the best method is to prevent the disease by using clean seed material. But once you have the disease in the field, can the disease spread via the soil?
Yeah, so for both ones, the Streptomyces is actually a soil-borne disease. So it lives in the soil. And once you get that, bacteria, it will live there for many, many years. And managing it can be complicated. And with
especially with the spores. Sure.
With the spores, yes, they can live up to many years. And with the Dickeya and Pectobacterium, they're a seed-borne disease, so they will not stay in the soil for many years. But they can, like you said, you mentioned, they can stay in the seeds, in the potato seeds, and the life cycle can move on for that. I mean, if you have a disease seed and plant in the spring, you will get a disease again.
So when you see the beginning disease, let's say from your drone, what do you do?
There are different practices. If it's with Dickeya and Pectobacterium, if it gets basically eradication would be one. You want to stop the spread, especially in the larger fields. So you want to stop, and you can eradicate as much as you can, but I mean, it depends of the spread, and it depends on how severe is the disease. But there is, as I showed you during the growing season, again, once you see it, it's there's some recommendation. You don't want to over irrigate and splash the bacterial spores from one place to another place, obviously. Again, there's not much you can do once it's there. Besides eradication.
So it's all preventive.
Yes.
You can do. Okay. Thank you.
Thank you. Thanks for your question.
Thanks very much. So staying still in the same forum before moving to the chats. Next up is George Burt. Kindly unmute and ask your question, please.
Well, good evening, Saltanat. I greatly enjoyed Can you hear me?
Yes. Hello.
Good evening, Saltanat. I greatly enjoyed your seminar. I have a question for you about the Scab part of your presentation. I want to make sure that I understand the results of your research. For as long as I can remember, I've heard people discuss whether or not one species causes surface scab and another species results in like a pitting scab. Okay. My understanding of your research results is that at the moment, it's not at the species level that we associate these two types of symptoms, but it is at the cluster of genes level that you associate with the different types of symptoms. Is that correct?
Yes, that's what we found in the research, and it was, the research is still continuing. Dr. Hammerschmidt's lab is still working on more isolates, because yes, you are correct. That previous, I guess, knowledge was that the different Streptomyces species were causing different lesions or, pitting symptoms. But in my study, both with turnips and potatoes, we found that it depends on the gene clusters that particular Streptomyces species carries on. And yes, so that's correct.
And I guess while I have the floor, I'll just make two comments. One, of all the different crops and diseases that I work with, certified seed production for potatoes is probably one of the best disease preventative measures. And then in addition to that, with Dr. Bruno Basso, we're doing a lot of work with remote sensing these days, not only drones, but also fixed wing aircraft and satellites. And what we hope to publish on in the relatively near future in relation to potatoes will be a new soil health concept, soil health indicator entitled thermal stability.
Thanks for sharing. I'll be looking into checking that out. Thank you. Thanks for joining the seminar today.
Thanks again. And one last raised hand so far. Kareem Moradia, please unmute and ask yourself a question.
Thank you very much. This is not a question, but just a comment. I just want to congratulate Saltanat for the very nice seminar. And we are very proud of our partnership with University of Central Asia. Saltanat came to Michigan State, did her master's degree and her PhD degree, and she's gone back to Central Asia, working with UCA and serving the region. And that's really was our goal, to bring young minds to Michigan State, and then they graduate here, acquire knowledge, and go back and serve their country, their home country, and the home region. So congratulations. We have an MOU with University of Central Asia, Michigan State University and University of Central Asia. And we look forward to renewing that and expanding. I'm in touch with Professor Salim Sumar, and we are looking to expanding our collaboration, training of your young scholars and also exchange of faculty and students. And I've been to University of Central Asia campus in Nareen. I would like to visit someday to Khorog. Once I tried to come, but because of the weather, I couldn't come. But thank you again and congratulations.
Thank you very much, Dr. Karim. Thanks for joining, and definitely we'll try, start some collaboration with MSU, and hopefully one day you'll be able to visit Khorog once this pandemic will be over. Thank you.
Thank you.
Indeed, I join also in the thanks, and we'll be delighted to host you here. And now moving to the chat, I see Professor Mohsen asked the question in, but he had to leave because there's another meeting. So here's the question. Some potatoes we use have parts with changed color. They're not totally destroyed and the tissue is still hard. Has this anything to do with disease, are you talking here? Or are these slightly darker parts harmful if one eats it? And Solmaz is seconding this question, saying especially potatoes that have gone slightly green under the skin, is this caused by toxic pathogens?
I guess with Dr. Moshan's question, did he ask when the potato has been cut? Right? Did you mention that?
It's nothing about the cutting part here. It's just as some potatoes we use have parts with changed color. They're not totally destroyed and the tissue is still hard. Has this anything to do with disease you're talking about?
It's very, very generic, I would say, and I can't really just, I guess, from this description tell, but basically, I guess.
How about we ask Salmaz, because Salmaz is seconding the question here.
No, it's just a funny question because, there is so much, I think, sometimes myths around vegetables and especially potatoes, when to eat it, how to eat it, what's the healthiest way. And especially, when they change color. And I've often heard people saying, when the potato, starts to go green under the skin, this is a bad sign and you should just discard it, or you should cut out those parts and not eat them. So can you put an end to this myth? Is it a myth?
I guess with the greening part, I can maybe help you with the myth. The greening potatoes, yes, they are not good because the green once the potato exposed to the sunlight, it does produce this toxin, which is not a pathogenic toxin, but it just produces these glycolloids that are not good for humans. So green potatoes are not good, definitely, and that happens when they get exposed to the sunlight. So you don't want to keep. Sometimes when you're walking in the field and the soil maybe gets washed away, and then some tubers become the sunlight can get through them, and that's when they get green parts. But in terms of general, I guess, recommendation in terms of potatoes, you saw the diseases, right? With the scab especially is the last one, the picture that I show you, different severity. Obviously, the last one you were not able to eat because there's not much left tissue. Like there's all deep, very, deep lesions. But with the rest of it, where you had, several, the scab, the surface lesions, obviously you can remove it and eat it. It's not pathogenic to humans. And same thing with other diseases. If the potato you are looking and you see some diseases, you can always, clean around and eat it, unless if it's smushy, like the soft rat makes very smushy, and it's not edible because the tissue is very soft, and obviously you will not be able to eat. But otherwise, the green potatoes are not good. Don't eat them, and the other ones, if you can just try to cut those areas, you can eat them, because the pathogens that cause disease in potatoes are not harmful for human, and most of it I know. So There are some pathogens of other diseases, wheat and other things that produce toxins. Those are not good, but with specific potatoes, they're okay to eat.
Thanks, Saltanat.
You're welcome.
Thanks so much. I don't see any other questions either in the chat or with raised hands. So I guess we've saturated, and we're also over our time. I suggest if anyone has any further questions or follow-ups to reach out to Saltanat after this call directly. Before we conclude this session, I would like to remind everyone about the next week's lecture by our colleague Nurlan Torokeldiev, and the talk is titled Evidence of Selection Oleosin-like Gene and Association with Environmental Variables in Persian Walnut Species. So we move into the trees next week. That sounds very interesting. Thanks very much again for everyone joining the talk. Thanks so much for Saltanat for providing such an engaging and interesting talk, and obviously highly relevant for food security in agriculture. And hopefully with the new facilities coming in, we'll be able to do a lot more and hopefully help the farmers around the region. With that, we will end the session today. I know Saltanat wanted to connect with someone here, but I'm not sure what will happen if I end the talk. Will everything disappear or not? But I guess just wanted to say that if you wanted to reach out and continue the conversation, perhaps you could connect separately. Is that okay, Saltanat?
Sure, maybe I can, yes. If you end, I guess it will end, right? I'm sure because you're a host.
Yeah, that's what I feel like.
Okay, maybe Nikolaus, I can call you separately on Skype or something.
Yes, Skype
Yes?
is okay.
Skype is good. Would you please email me your Skype account, the ID?
I'll send you a mail. Yeah.
Okay, thank you so much. Thanks, Muratbek, for being a moderator for this talk, and thanks everyone for joining and listening. And as Muratbek said, you can just type my name if you have any questions. Find my email. I guess I didn't put my emails in the talk, but I'm more than happy to answer anyone's questions or myth, like someone said. If you have about potatoes, potatoes are the best, so you can eat them. They're good. All right. Thanks.
Okay, thanks so much. I see Saltanat that Solmaz already put your email in the chat box. So everyone who wants to connect, here it is. All right, so have a great weekend ahead, and we'll connect next week.
Bye, everyone.